computer and share screen. Well, there we go. There it is. Everybody see that? Yeast, laboratory diagnosis with yeast. So, you know, this is the one we're going to encounter the most in my with our patients. Maybe not in micro itself. You know, I was working Saturday and ended up walking in and finding a sample on the table, and it had fungus culture. So that was the order. And so I went into the refrigerator, and guess I was looking for, you know, my Sabernown plate to plate that out and get it growing. And then I read on my procedure list that was a send out. So we're going to talk a little bit about how that works, but that was a ruptured ulcer. So a place that does some surgery ended up getting some fluid collected from a ruptured ulcer, and collected it in a sterile container, sent it to micro to be grown out for fungus. So most of the time you'll encounter yeast in urine samples, urinalysis, and you need to make sure that you don't confuse that with white blood cells. So you'll see yeast, and you may see budding yeast. You may see more than just budding yeast. But they do have definitely with unstained in a urine, they do have a tendency to be confused with white blood cells. So keep that in mind, and keep your result from your urine dipstick in mind too, because if you see leukoesterase indicated, and then we walk into, or not indicated, then you walk into a sample that looks like it's full of white cells, probably doesn't add up for white cells. It'll add up for yeast. But that is probably the most prevalent place you'll find it on your patients, is with the urinalysis. All right, so yeast infections. We have, what do we use yeast for, right? So we enjoy yeast because we can use it for bread, making bread, and also beverages. So helping fermentation to produce ethanol in our beverages. Those are the beverages I'm talking about. Our beers would be one. And then we see with biofuel and ethanol. And right now, you know, we've had a kind of a break from biofuel. Not as much push for biofuel. Not to get too political, but I don't know if you've noticed, but something's going up right now, and that would be the price of gas. So even before a new administration takes over, we're starting to see increase in gas. When gas goes up, biofuels maybe come back into the picture to go to a different route, you know, for energy. So two that we start with is Candida albicans. I think we've seen that one. You grew that on a plate. Cryptococcus neoformans, those are two medically important, causing wide range of infectious diseases. So these are some of the yeasts that we need to focus on. We got plenty more names to come, but we'll start there. What causes a yeast infection? Well, the same list that we've had all semester, it seems like: cancer chemotherapy, radiation therapy on steroids, antimicrobial agents, right, going after a treating of an infection and then setting it up for an opportunistic yeast infection to take over, kind of along the same lines as our C. diff thinking. Metabolic disorders, diabetics, yes. Okay, pregnancy, yes. Menstruation, contraceptives, malnutrition, uremia, definitely all putting the patient at risk for yeast infections. And then finally, immunosuppression, whether we're doing it for help the patient to keep a transplant intact, or if it's an autoimmune immunosuppressant. So yeast play that role. Fungema refers to the presence of the yeast in the bloodstream. It has disseminated there, and it may cause death, and it's not uncommon for it to cause death in a cancer patient or an AIDS patient. So we definitely will have that. A brother of mine ended up with a surgery where it didn't go well. They were removing some of his large intestine, and he had to stay open. His wound was open, it wouldn't heal. So he was laying in a hospital bed with an open abdominal wound, and one of the things he ended up with was a fungus infection. So definitely can play a role in surgeries and wounds. Yeast sometimes colonize the mucosal membrane, causing disease. And in these cases, they're recovered in low numbers and mixed cultures. So I'd be surprised on that sample I drew up. It was probably 10 cc's of body fluid. Definitely could be colonized with fungal, but they want to send it out and make sure. And we're going to talk about what maybe I could have done in my micro if I'd had the capability of doing it, where you actually can take on a little more of the ID for a fungus. In invasive infections indicate when the yeast are recovered from sterile site. This is where we think about like body fluids or tissue specimen where they're not supposed to be. And I think of cerebral spinal fluid. You do a cerebral spinal fluid and you look at that and it looks like it's got budding yeast or hyphae, whatever way you find them. Definitely not supposed to be there. And we think of that with pseudohyphae and hyphae, often seen in tissue, low oxygen levels, and invasive disease, Candida albicans is definitely can set up that way. So some pictures. So now we'll take on some names. Blastoschizomyces capitatus, and of course it has a otherwise known as Trichosporon capitatum. So it's an opportunistic pathogen, which most of these are, associated with disease of immunocompromised host. Candida albicans, that's the one, if we're going to remember one, we're going to remember this one, right? May colonize the mucous membrane of the GI tract, urogenital tract, upper respiratory tract. The yeast is present in vaginal mucosa of 5% of all females. So it's definitely present. Candidiasis, ACEs, right? Candidiasis. And we're going to go, anyway, infection caused by a member of Candida. We see one in particular called thrush, invasive fungal infection of the oral cavity. We see this with infants where they get thrush, and then we have vaginitis, balanitis, sexually transmitted infection. But this is thrush. Okay, so you look at the back of the throat. I see a tongue and a uvula there, and you see that this is all yeast growing. Okay, so how do you cure that? You give the patient drops, right? Does anybody know the medicine? I mean, we had to do this for ours too. I can't even remember. What is the medicine? What drops did you get prescribed? I guess Heather's the only one that can talk to. Heather, y'all have thrush?
No, thank God, neither one of my kids had that.
Okay. Usually you think about, bottle caps, passies, anything they're constantly, putting in their mouth to introduce that into their throats. Here's a cervix, same thing, candidiasis. Thus, hopefully I know you hadn't been to body fluids, but that's epithelial, these big skin cells out here. And then what we're looking for is these kind of structures, a budding yeast here, definitely here. And you can see this with a urinalysis. I mean, you'll see it. You're not going to have to do anything special. You're not going to have to stain it. You just need to get good at, there's another budding yeast. just getting used to looking at it. And
Bednar
taking,
has a really cool slide of a bunch of yeast.
okay.
Your analysis.
But I did have what was, you know, I think I shared this story too about the clinical site asking me, did I teach this? You know, they're like, well, our students, your student came to clinical and looked under the scope and said they've never seen yeast under the scope before, right? So here are some slides. You know, of course, we're staining the hyphae here. And then, of course, I think that's from your test group. I remember that one's very familiar. And I was thumbing through Mr. Payne's micro book the other day, and of course, all these pictures are straight out of his text. He did the chapter on fungus, and he used all these pictures. So, chest X-ray. If you can't remember what that looks like, heart, depression in the middle. Definitely got some spots going on. Endocarditis, invasion of the cardiac muscle here. A lot of hyphae stain from there. So Candida albicans, once the patient has like tetracycline or aminoglycosides, those are antibiotics. They disrupt the balance of the microbial population, and this kind of sounds so familiar to C. diff, right? So that competes, the Candida, the yeast, competes for the binding, but when the bacteria is knocked out, then it has a spot. So the binding and the nutrients, so this yeast, which is resistant to antibiotics, is able to proliferate after treatment of tetracycline aminoglycosides. So microscopic morphology, here's the switch. You know, we go from our Savin-Hour, culture over to cornmeal agar to get it to change into the Blastoschizomyces, pseudohyphae, true hyphae. And we tried that, of course we did Candida albicans, didn't quite see it when we did it in lab, coming off the cornmeal agar. So we did see a lot of, the regular circles. We didn't really see these hyphae and pseudohyphae. So our next one is Cryptococcus neoformans. Of course, these are our two, pathogenic, more likely to be found in our patient than the others. Fungal disease, Cryptococcus is a fungal disease, inhalation, okay, but may affect many organ systems, so it's primarily in the pulmonary. So we don't think of Candida as inhaled, we think of it as vaginal, thrush in the mouth, in areas of that way. But Cryptococcus is going to be inhaled. Immunocompromised host, immunocompetent host again is asymptomatic and very mild. So normal, hey, no issues. It does have a predilection to be a central nervous system. So it does move to the central nervous system. And this is what sets up the meningitis or brain abscesses. And they are poor prognosis if that ends up happening. Isolation of the organism or detection of the capsular antigen. So we can do a latex agglutination for that off the cerebral spinal fluid. The habitat, pigeon poop. Okay, so pigeons, kind of like blackbirds, they like to huddle in big groups. They like to find areas like under overpasses or in, buildings, around buildings, awnings, and then they poop everywhere. So you definitely, they habitat there. So the Cryptococcus neoformans is now in the poop, wherever that poop was. And if it's in a building, up in the rafters and you come in and, you're inhaling that. So you can definitely inhale those. Polysaccharide in nature is an anti-phagocytic. They're capsulated and they're microscopic. They're Blastoschizomyces. Look occasionally rudimentary pseudohyphae. So definitely capsulated, a little thicker. See a capsule, kind of faint capsule around at least this one. This one, So the capsules don't stain very well. This is from a liver sample. Lungs of an AIDS patient. That looks like an India ink. Candida glabrata. Opportunistic pathogen, colonizes GI tract, skin, upper respiratory tract. Infections are urinary tract infections, or fungemas, that's fungal in the blood. Glabrata is the second most common cause of candidiasis after albicans. So albicans comes first, then glabrata. The tissue specimen, glabrata resembles histoplasm. What is the H again? Histoplasm. What is that? Plasma capsulatum. Is that correct? What is the H for capsulatum?
I think it's histoplasma.
Okay. Yes. Microscopic morphology, small Candida, and only no pseudohyphae here. Trichosporon ovoides. This is a white hydra. I think we've already touched on this one. Widespread in nature, occasionally on healthy skin. Microscopically, we're going to see hyphae, pseudohyphae, true hyphae, arthrocanidia, and blastocanidia. Geotrichum species. Rare infection that may produce lesions in the mouth, intestine, and lungs. Did somebody have a question? I hear somebody. So, okay. Opportunistic pathogen, again, immunocompromised host. In the sputum, we have gram-positive arthrocanidia. So we take that sputum, and most places, most micro labs require you to gram stain all sputums, all wound cultures, okay, all blood cultures that are positive, of course. So you'll be getting used to gram staining sputum, and what we're looking for, we may find here, right? And we may find some arthrocanidia that, and hyphae from our gram stain. This is like a gram positive, looks like just what we would find. And you may be new to sputum, and you may be doing this and going, that's just part of sputum, right? Something just stained purple. It's really not bacteria looking, but it's purple, right? So hopefully you'll get some experience with this eventually, where you do stain sputum and see what sputum has in it for you for the stain. Again, white hydra. So question was, when I did my fungal collection, the specimen was collected Saturday for me to send out to Quest. And so, as the good micro tech I am, I wanted to go make sure I wasn't going to do something wrong for Quest, because Quest will reject things. And it's frustrating if you goof up something or don't send something to them and they just reject it, and you don't get it back. You don't get a retry. So part of our specimen processing Saturday was to put on there what was on the label. It came out on the printout, was irreplaceable specimen, which meant we're not going to be able to go in the fridge and get a blood tube with more serum to send you again to Quest if they deny it or they reject it. So that is key for processing, and you'll get experience with Quest and LabCorp and AEL, depending on where you go. There's always going to be a reference lab option for hospitals and clinics. So part of that was it was irreplaceable, but my question was, where do I need to, do I need to do anything with it? So some of the prep was, if it wasn't a body fluid, say it was some other tissue, you'd want to, centrifuge it down. But this was pretty easy. It was basically label it. It was in a sterile container and put it in the fridge. Like we would do most of our specimens. We either freeze, room temp, or refrigerate all specimens sent out, whether it's blood or body fluid. Sometimes they ask you to plate. They'll ask you to go ahead and make a plate, or you got initial growth. And if you had that, then you could send the plate in to be referenced out. So yeast are routinely isolated from sputum. Colonization of the oral cavity, we saw that with thrush, and may be seen on a gram stain or culture media. So we may culture something out and look at it the next day, and it looks different. It has a different look to it. They take a little longer to grow, so it may not be the first day that you see it, but it may be the second day of the growth that it actually shows up on. Urine should be quantitated. As for bacteria, small numbers of yeast are insignificant unless they are one of the systemic fungi or the yeast phase. So usually on a urinalysis, we'll just say yeast seen. That's it. We're not going to culture that yeast. We're not going to identify that yeast. We're just going to let the doctor know that they need to be put on an antifungal instead of an antimicrobial, where they won't be getting, Rocephin. They'll be getting, antifungal medicine for their yeast infection. If we have cerebral spinal fluid, that's where I was saying we'd want to spin that down with centrifugation to concentrate it and then plate the sediment. So if I'm not setting it up, like I wasn't setting up that sample Saturday, I just sent the whole sample to Quest. They'll do the centrifugation, and then they'll plate the sediment. And remember, the sediment is what settles down at the bottom of the centrifugation. So remember that. Don't spin it down and then plate off the the other part. What's the other part? Solvent versus solute, right? Solute is considered the settlement. India ink can be used to demonstrate encapsulated yeast. If we had Cryptococcus neoformans, we could use the India ink. Well, there you get a good look at Cryptococcus there with its capsule around it. And I think sometimes these would look really good on my wall. Like if I could paint that, all right, that'd be a great painting, right? Everybody come over, y'all would come over sometime, you go, hey, Mr. Reck, that's a cool painting of Cryptococcus you got on your wall.
That's what we said about the Candida albicans picture.
Yeah. So we can also do latex agglutination. You're very familiar with that if you're in immuno. Used to demonstrate the capsular antigen for Cryptococcus neoformans. So we look for agglutination there. And then a direct exam is really key for specimen processing. Gram staining, we can use acid shift stain for tissues, chalcofluora white, and potassium hydroxide for skin samples. To culture it, we put it on our Savaron dextrose agar, our CDA. We can use brain heart infusion here. We had some of that going for our research project this past semester, where that's what we used. That was our choice for growth media. A TSA is always around, and it's enriched with sheep blood. So that's our normal blood agar plate, is TSA with sheep blood. So you can definitely go ahead and plate it on sheep blood. But as you saw with Savaron, what we like about that is it just looks different coming off that than it does blood agar, but it still will come off blood agar. Cyclohexamide modified Savarons, which inhibits the saprophytic fungi and Cryptococcus neoformans. So if you wanted to go, definitely could modify the Savaron to be real selective on what fungi grew. Then switching from Sabaron to cornmeal like we did in lab. So we went through this process. We can be used to demonstrate the microscopic morphology of yeast. So hopefully you appreciated our cornmeal agar, taking the slide, the cover slips off and staining that with our lactophenol cotton blue. Germ tube tests, sometimes those are available. That's more of the reference lab. Going to use the germ tube test where you incubate the test organism using a half ml of serum for 2 to 3 hours. You look for tube-like structures emerging from the Blastoschizomyces. Should be parallel walls or true hyphae, no constriction point, or pseudohyphae. It's supposed to be pseudohyphae there. There's no constriction point on those hyphae. So biochemical test, you know, we didn't get the chance to use the API-C, but there is options for identifying these biochemically. Assimilation, aerobic utilization of carbohydrates. We can do fermentation, anaerobic degradation of carbohydrates produces our ethanol and our CO2. Nitrate utilization and urease production. So there's not as many biochemical reactions that we go for with C, but they're there. So how does the lab diagnose fungi? Direct exam. As a group, they grow much slower than bacteria. So that reason a direct exam in some cases is better. You know, if we could go ahead and get that done, if we can get it on there and kind of say, hey, we think we have a fungus here or a yeast, whichever way you want to call it. But if you send it out, they're going to keep it for a while, right? Up to 4 weeks. The presumptive ID for pathogenic fungi may be based on direct exam. If you're good at that, characteristic microscopic morphology. I know you're going to be in there one day and you're going, man, if I could just have my access to my Blackboard micro account for lab, I could look over these slides one more time. But there's definitely atlases out there. You can also search. But hopefully you'll remember some of the ones we're going to test on Thursday. So as you see, here's our big test, right? Characteristic microscopic morphology of pathogenic yeast. So I need 4 volunteers. That 4 that have been studying their slides. So we see dermatitis here, large scale single budding yeast with a broad base. And what does the B stand for?
Isn't it like blastomycosis or something?
Myces, yeah, I'll take that. Blastomyces, that's on our list. That's number 76, our slide there. Braziliensis. Who wants to take that one? Somebody other than Allison. Multiple budding yeast and has the pilot's wheel. I know we've seen that one. I think we saved that one, right? So who's our Braziliensis name? Anybody? It's not that hard. Come on. It's slide 81. Go ahead. Who's that? Who's Heather?
That's me.
Oh, okay. Go ahead.
It's very hard to say.
No, it's not.
It's paracoxio. Nope.
You got it. I mean, you got it. It's right there. Paracoccidioides.
Oh, there you go. That one.
Oi. We gotta do oi. Oides, right?
Just, yeah.
That's slide number 81. We have that one on our list.
Mm-hmm.
We got C. amididus. and that is spirulums of endospores. And what does the C stand for? It's not para, so what would it be? Somebody other than Allison and Sharuk. You know, I hadn't made out the test Thursday. We can always go to. Can you write in Blackboard? Can y'all write and type answers in? Can I just put a blank and just say type it in?
Please don't.
Well, somebody needs to help me with a name. It's not para this time. It's just
Is it the coccidioids?
ovoides. Yeah, coccidioides again. Thank you. Okay, that's 3. And then we got one we've already visited this lecture, which is capsulatum, small oval intracellular yeast. But who wants to help me with the H? I was looking for,
Histoplasma.
yeah, histoplasma, that's 68 on our list. So all 4 of these are on our list to recognize from a picture, but also to put in our, like, that's the way you do it, right? Direct, slide says direct microscopic examination. So how do we go about getting that direct? We do potassium hydroxide prep. A micro lab has that, has a bottle of it. Digest the protein, therefore it clears the keratinized tissue so fungal elements can be visualized more easily. The chitinous cell wall of the fungi is more resistant to alkalis, but with time the fungus will also disintegrate. We can use the periodic acid shift PAS stain. Hyphae and yeast cells stain brilliant magenta color while the background is decolorized. And I know we have magenta was one of our colors from lab that was not universally recognized in translation to Chinese. So somebody help out with magenta. What color would you compare magenta to?
Purple and red.
Yes, thank you. Purple, reddish purple, magenta. Acid fast stain we can use here to detect ascospores of the yeast. That's magenta, right? Reddish purple. India ink prep to visualize the hyaline of the Cryptococcus neoformans. India ink does not stain the capsule, provides a dark background for visualizing the capsule. So again, I think we saw one earlier. It was hard to kind of see, but it did. There was one that was great picture we're putting on the wall. The gizma stain or right stain can be demonstrated with histocapsulatum, histoplasma in bone marrow smears. So definitely we talked about areas that we wouldn't want this to show up, and all of a sudden it's in your bone marrow. Not good, right? Fluorescent antibody stains, where we take the fungi and tissue and body fluids and we attach a fluorescent antibody that attaches to the actual fungus. And the reason why we're not spending a lot of time on these is because we're going to come back to that slideshow at the end today, like we promised. Gamori, methenamine, silver, GMS stain. Again, another stain. Fungal elements such as budding yeast and hyphae will be outlined in black, while the internal parts are deep rose to black color. And this background will stain green. That's our Cryptococcus neoformans we've already seen from a lung from an AIDS patient. And what's this one? Pneumocystis what? No, we've been practicing this name. Remember, I know it's we. Wero. Oh, and then what? How's it finish? Somebody?
I say Girovecchi.
Girovecchi? Yeah, but we
That's
had a.
just how I say it.
I know. I've forever called it Girovecchi, so I wouldn't forget how to say it.
Mhm.
But we had a pronunciation last time or time before on our slideshow. Anybody remember what that was? How to pronounce it perfectly?
Is it Yeasts?
That's it. Okay, thank you. Culture procedures. Primary isolated media must provide a carbon source, so we put some sugar in there, and a nitrogen source, so we put some peptones. Blood or body fluids are added as an enrichment. So our Sabaron dextrose agar, or CDA, contains 4% glucose and peptones with a pH of 5.6 to inhibit bacteria. So that's why you would want to switch to Sabaron instead of TSA with blood agar, right? Because if you get bacterial growth, it may confuse you. So Sabaron, the reason I was looking for it Saturday was because I wanted to plate. I could have streaked that out and maybe seen something either Sunday or Monday to let us know, maybe indicate, yes, fungal elements present, or, presumptive positive. As it is, I have nothing. So I just sent it off, picked up on Monday. Who knows when that result will come back. Emmons modification, 2% glucose, pH a little higher, 6.8 to 7. Chloramphenicol and cyclohexamide, which is, antimicrobials, inhibit bacteria and saprophytic fungi. So that's our enhanced Sabaron dextrose agar. Brain heart infusion, we could use that where we use cornmeal. For us, we use this for the conversion of the mold phase to the yeast phase. Inhibitory mold agar, recovery of the fungi from clinical specimens. It contains chloramphenicol, again, to knock down the bacterial. A birdseed agar. I don't know if we've mentioned birdseed agar. I've never seen birdseed agar, but also known as cafetic acid agar, Niger seed agar, and that is for isolation of Cryptococcus neoformans from a contaminated specimen of sputum, because a lot of times sputum just looks when you plate it out, and a lot of times we don't see what we want. It's just a bunch of flora, and a lot of sputums get discarded as flora, where maybe if we were able to take that and put it on a specialized agar instead of just blood agar, we'd see, we wouldn't miss some of our fungal friends on a sputum. Converse medium promotes phylum formation of Candida immitis. Is it immitis? Is it Candida or is it Cryptococcus immitis? Which one is it?
It's the Coccidioides.
Oh, it's Coccidioides again. Yes. Thank you for, I figured y'all would throw things at me. And then finally, to kind of leave you with emerging pathogens here at the end. Some that we may not have heard of yet, and Candida auris. Emerging pathogen, yeast can cause serious infection. Hospitalized patient, frequently resistant to multiple antifungal drugs. So when we say emerging pathogen, that's why, because we think, oh, it's just a yeast, we're going to treat it with antifungal, and it doesn't go away. It's difficult to identify. can be misidentified without specific technology. Mis-ID may lead to inappropriate management. It has caused outbreaks in healthcare settings. For this reason, we want to quickly identify auris in a hospitalized patient. Candida dubliensis, dubliensis. First described in 1995, very closely related to albicans. It has been implicated in oral candidiasis with HIV positive people. It's been recovered though from HIV negative people with clinical signs to oral candidiasis, and from the genital tract of some women. Phenotypically, it shares with albicans, producing germ tubes, chlamydiospores. So it's going to be, I don't think you're going to be sitting on the edge of your seat going, I need to make sure this is not dubliensis. But if you're in a hospital and you start having trouble with surgery because everybody's coming out with fungal infections, maybe you've got, some sterilization procedures of your instruments not going so well. And then finally, Phylum Microsporidia. Again, we may not think of this as fungus as being parasites, but they are obligate intracellular parasite. Reproduction involves formation of minute spores ranging in diameter 1 to 2 micrometers, and we'll get into that more in parasit, that have polar tubules and filaments. The tubules are used to inject the infection material, the sporoplasm, into the host cell. So, evil little thing. Epidemiology, the Microsporidia have a wide host range, including all classes of vertebrates and most invertebrates. They produce significant disease in honeybees, silkworms, fish, laboratory rodents, rabbits, other fur-bearing mammals, and primates. There's 13 species of Microsporidia that infect humans. They give human disease such as chronic diarrhea, wasting syndrome, keratoconjunctivitis, pneumonia, bronchitis, nephritis, urethritis, prostatitis, hepatitis, encephalitis, peritonitis. Let's just keep on giving the itises, right? There's 5 genera that have been reported in humans. These have names like encephalolitozone, Trachyplestophora, Nosema, Vitiforma, and Enterocytozoon. Most common disease is intestinal infections of AIDS patient, diarrhea, wasting, and dehydration, losing several liters of liquid feces a day. We're going to biopsy feces, urine sample, respiratory secretions, other body fluids. We're going to use a modified trichrome stain, looking for spores stained pink red with clear polar central zones. Other staining. Here's our CDC, basically a parasitic life cycle. Look to it. Spore here. Invasion there. All right. You made it. Yay. Just going to stop the share here. Any questions on that introduction? Basically, that's what we did here. We introduced mycology. it is part, but not a, you know, you're going to spend 90% of your time in bacteriology anyway. Mycology, parasitology, virology, you know, all those ologies there at the end, they make up the other 10% probably, if I was weighing out your time in micro lab that you'll be spending on your patients. All right, so that is that. So that is our last lecture. That was, oh, what lecture number? 30, no, 24-ish, right? Lecture 24. Okay. All right, so now we want to switch. We'll switch gears. Oh, and I did want to remind, I don't know how many I have on this call, that you got a email from Dr. Walls to double check your Micro 2 registration. Remember that if some students are required to be in the section that says lab assisted, or lab A, that class is going to require you to come to campus randomly through the semester. The other Micro 2 is all online, so requires no time on campus in that one. So double check. I looked at the roster today. It looked like a couple of students needed to switch out on sections, so double check your sections. And I'm talking mainly to our friends from China. Double check your registration and make sure you're in the lab assisted section for the class. Okay, all right, good deal. All right, so now we're gonna go to Blackboard and look in the lab, and we're gonna pull up our PowerPoint mycology slides, and we're gonna review at the end. But first I wanted to review the syllabus for what else was there. Or like, oh, is it just not gonna be mycology slides, right? What else could there be in Medical Micro 1 lab? And I think, what did we do? We did an unknown at midterm, right? So what we did, you did the unknown and you ID'd it and you did your test, and then we graded off, did you do the right test, the wrong test, that kind of thing. Well, what we're gonna move to, we can't do that this time, but what we can do is we move into susceptibility testing. Don't forget that. So that's the main other part of this section of micro, where we looked at antimicrobial, the MIC, major inhibitory concentrations. And the main thing we worked on was, is it sensitive? Is it intermediate? Is it resistant based on where the growth was? So you can see some examples that would be easy to put into a Blackboard exam, where I gave you growth in certain levels of antimicrobials with a key, not a key, but a choice of where we're going to call it sensitive, intermediate, or resistant. So those are the kind of things that definitely can put on into there.
So is that information in our lab packets already? Or do we have that?
You have that with the handouts, with the gram-positive combo and gram-negative combo handouts. That's where you'd find that.
Like the big diagram thing?
Yeah, well, it's a big it's let's see, where's my stuff? Didn't I print off something that looked something like that? Didn't I print off a handout for both of those, how to set them up, how to read them, procedure manuals?
So I have dried gram-positive procedure manual, and we also have like the same one, but it's for like gram-negative.
Yes.
We have, yes, those are the two that we have, and the mold slide culture techniques.
Okay, and then I gave you a handout on how to read them, right? Let me get it. show it.
Yes, please, because I don't remember having that one.
Did y'all get these? Y'all still have those?
Didn't you collect those after call?
Yeah,
Um,
then.
yeah, I might have.
I have one still, but how are we supposed to study that?
Well, that's it doesn't really tell you.
Is it more of like you should know what to mark if the specimen isn't there?
Yes,
Is if it's heavy?
yes. So the choice would be like 3 circles of the different levels of antibiotics. So it would be, you know, it's like 2, 4, 6. Okay, or let's just, yeah. Well, it would be 2 circles. Let's go 2 and 4. There's growth in 2 and 4. Is that a resistant strain of E. coli or not?
Yes.
Okay, so if it was 2 and 4 and there's growth in 2, but it's clear in 4, is that a sensitive, intermediate, or resistant strain of E. coli?
Intermediate?
More than likely, yes. And then if it's not growing in either 2 or 4, is that sensitive, intermediate, or resistant?
Sensitive.
So that's it. I mean, that's
Okay.
basically what I'm adding in, that kind of thing. I'm not going to ask you, what does AMP stand for? Is that ampicillin? I'm not asking you that kind of stuff. So it's just basically that kind of check, just to add a few questions to the 38 slides that we have coming that we're about to look
But
at.
seeing if we can just read it, like if we remember how to read the panel, kind of, but in a.
Yeah, and it won't be just a vague question. There'll be more to it. But just to check your, you know, competency on reading MICs. That's what I want to check. Not that, you know, not that you know, but just can I give you examples of where growth is and where growth's not, and you read through that, can you tell me, interpret that? So that's the main thing I want to check before we move on to micro 2.
So is that what is also going to come out of the microscan papers for the positive and negative that you gave us? Or what was coming out of those?
Yeah, that gives you more of what the panel has on it and the levels of each one and what that means. You know, like when you look at it, if you were studying the manual, and it gets into the point, I think, let's see, take control, let's see. It gives you all the different lists, and then it tells you to do. I'm looking for exactly where it is. Give me a minute. Well, I thought I just saw it. The last, no. It may not be worth going into those. I'm just looking at it. I mean, it gives a little bit of an example, like the cefoxidin screen, negative and positive. And then oxacillin talks about that. I'm looking at the gram positive. But for the most part, there's a big list on the 1.6 that says antimicrobial agent, susceptible, intermediate, and resistant. And it gives you every one of them. So that's the only thing I can see that you would want to like kind of look at. Everything else is how you develop it. So. That help?
Yes.
Okay. Any other questions before we go to the PowerPoint and go through our list? Have y'all already know all these, right?
I do have a question for like after that. Like, I just want to ask a couple questions about immuno again, but I don't want to like interrupt micro again. So yeah, would that be okay?
Sure. What's your question?
Well, it can be after we do the slides.
Oh, okay. All right. So we'll share my screen. If I can find it, there it is. Everybody see that? Right. Seeing it? Aspergillus. Got
Yes.
the Candida, the Phyllidias, the vesicle, the stalk, foot cell, remember all those? So I've got my trusty list right here that we highlighted and shared with everybody all the way to slide 109. So just kind of move through that one more time so we can kind of double check, make sure that I still have it right before I make up this exam. So this is one of the ones, this is the Aspergillus that I want you to remember. And it was so bad, like when I got off the call last week and I went back to that slide, I found one of these. It was like, it was so good. And I was like, I just didn't see it when we were looking at it, but there was definitely a really good Aspergillus head on one of the samples I looked at, the stain. But we'd already stopped the share and all that, but yeah. Then we went to 4, which is fumigatus. Then we went to 6 for penicillium. Fusarium, number 7. Acremonium, number 9. To 11, where we had Paciliomyces. (13) Scopulariopsis, sorry, Scopulariopsis. To 15, where we did Alternaria. To 17, which was Bipolaris. To 19, which is Curvularia. To 22, which is Mucor. To 27, which are spaghetti and meatballs. Malassezia furfur. To 30, which is a Hortea or Necki. Tinea nigra. To 38, which is our Microsporium canis. To 39, which is our Microsporium gypsum. To 41, which is our Epidermophytum flocosum. (43) Trichophyton rubrum. (44) Trichophyton Menogrophytes, right? Menogrophytes. "46, Cladophilophoria. 49, Philophorium variecosa. 51, Fonsesia pedrosoi. 54, sclerotic bodies. 58, Nocardia asteroides. 59, Pseudosporium apiospermum. 63, Sporothrix Skinecki. 67, we have two of these? Oh, the different form, right? Mold form. This is Sporothrix Skinecki. And then a lot of slides we just saw on this last slideshow from lecture was Histoplasma capsulatum, 68. So the mold phase of 72, which is Histoplasma capsulatum mold phase. '76, Blastomyces dermatitidis. 81, captain's wheel here of Paracoccidioides brasiliensis. To 88, Coccidioides amididus, endospores coming out of the spirulum. To 90, which is Coccidioides amididus mold form. '92, Aspergillus fumigatus. To 96, from lecture today, Candida albicans, gram stain.
I mean, wouldn't you put that on your wall? Look how pretty it is.
That's very pretty, yes.
Yeah, this one's gorgeous.
So if we put that at auction, how much would you pay for it?
I mean, I buy stuff like that cheap, so I probably wouldn't pay much, but I would buy it if it was cheap enough.
I was like, yeah, we had an auction going on for your scholarship endowments that we're raising money for MLS scholarships. And they had a Dr. Fauci bobblehead from the Washington Nationals game. And it got up there. It was at 85 last time I looked. Uh, 101 is Trichosporon species. 103, Geotrichum species. The 107 is Cryptococcus neoformans. Had that one today. That's from spinal fluid. And our last one is 109, which is our pneumocystis What was it again, Allison?
Yearovetsy.
Vetsy. See, I always want to say Becky, so it's Vetsy.
Yeah, like a vet and then Z-E-E, Betsy.
Okay, Vetsy. Pneumocystis Vetsy. All right. That's, how many was that?
38.
38. Okay. 38, multiple choice on those. So any other questions? I'm gonna stop the share again. Sorry.
How many questions is the micro lab test?
Well, we said, what was the immuno gonna be?
25.
25.
Mhm.
This one might be 25 too.
Okay, thank you.
Yep. I don't plan on doing all 38 slides and finding 12 more questions to put on there. So I like 25 for lab, 50 for lecture. Any other questions? Any others?
If not, I do about immuno.
All right, shoot with immuno.
Okay, so for example, the status strip A papers, I have, like, I'm currently making a quizlet for it. I have like the intended use, summary and explanation principle, and like the procedure, but there's like a lot of like external quality control, internal procedural control, all of that I can like not, okay.
Yep.
What about limitations?
Nope.
Sweet.
No. Right now, you just need to know what you're testing for, why you're testing it, and how you're testing it. That's kind of the three things.
Okay.
Okay, reading some chat between you. Anything else? All right. In our Zoom tradition, we need a final Micro 1 Zoom call post today. Make sure I get everybody. This is the big screen. Okay, getting there, yay, yay. Good. Got two more. Wooey and Zichong. Wooey and Zichong. There's, all right. 1, 2, 3, everybody. It's all over the test. Yay. All right, you've been a great group. We'll see most of you next semester. Remember, we're here all the time. I'm here till graduation. So if there's any questions after exams, make sure you come up here, or you can email me if you're not on campus. And we can Zoom out and answer any questions or email any questions you might have over the test. All right. Study hard. You got 4 tests in 2 days. You can do it. All right. Bye-bye.